Alright, just a quick video on like linked genes and identifying recombinants and that kind of thing. So let's get on with it. First off, a few definitions. Linkage doesn't follow Mendel's law of independent assortment. So if you think of it kind of like, you have allele A and allele B. Okay, independent assortment means just like probability. Where A goes doesn't influence where B goes because they're on different chromosomes. So B can choose to go here or B can choose to go here. If they were linked, where A goes, B would go, and where B goes, A would go. So they are dependent on each other. So if it's Mendelian, you assume genes are unlinked and assortment is independent, and that's often a point in a mark scheme when talking about this kind of thing. Linked means it's on the same chromosome, so naturally the chromosome is what moves. So if you have A and B on the same chromosome, this chromosome is what pulls to the daughter, haploid gametes. So they obviously move together, so they're dependent on each other. So linked genes means the genes are on the same chromosome. So there's one definition. Okay. Next definition you need to note is a test cross is something with a phenotype that is dominant. Okay, if we recall, phenotypes are things of what you look like. So a brown-haired dog has the phenotype for brown hair, but say a black-haired dog. Okay, so this dog looks black. You look at it and goes, yep, that's got black hair, but we don't know if it's like this or if it's like this. Okay, because both of these would look black, but we don't know its genotype. So a test cross is how we determine this. So we get something that looks dominant. So we know, okay, it's got a dominant phenotype, but what's its genotype? So to figure that out, we cross it with something that is known to be homozygous recessive, and how we get to the result I'll explain. So we know it's homozygous recessive. We cross it with something we don't know what it is, and then we look at the ratios afterwards, and we can find out whether it was a heterozygous or a homozygous dominant genotype, even though we knew the phenotype was dominant. So remember, phenotype is how it looks, genotype is it's these things, it's genes, and that is a test cross. Okay. Next definition we need to know is a recombinant. A recombinant is something, it's a genotype that does not appear in the genotypes of the parents. So I've written two kind of dihybrid genotypes of two parents here. We've got a dad and a mum, and let's imagine the kid ends up like this. Okay, just quickly check yourself. Is this a recombinant? 3, 2, 1. No, it is not. Why? Because it matches the mum. Okay, the mum is small a, big B, little b, and so that's what the kid is, and that's also what the mum is. These match. If we notice, these are the same. So the same means it's not a recombinant. Okay, a recombinant is something that is not in the parents. Okay, let's imagine a kid gets big A, little a, small b. Okay, quickly check yourself. Are they a recombinant? Well, this first part matches the dad, but then this little b does not match a parent. So this whole thing does not match a parent. So therefore it is a recombinant. Okay. Now that we've got the definition of a recombinant down, let's try this question. I just have to get rid of all my working on it. 1 second. My bad. All right. Now we can go through this question on how to identify recombinants and do it quickly. Okay. So we have this: an individual is heterozygous for two linked genes. Okay. My first advice is when doing recombinant questions, they usually always give you this horizontal representation of it. In my opinion, that's just really annoying. So I always write it out again, but vertically, because I find it's a bit easier to work with. Okay, so that is just that, but written out in vertical, because we can see the gosh, I've done that wrong. Sorry. Yeah, you're just twisting it like to be on its vertical side. So it would be big A, big B on that side, and little a, little b on that side. So that side and that side. Okay. Sorry, I was just messing around there. But now we've got that vertically, we can read the question. To investigate the frequency of crossing over, a test cross is carried out between the individual and another that is homozygous recessive. So homozygous recessive, they would be like this. So that's just, we need to identify that they're homozygous recessive. What are the possible recombinants in the offspring of this cross? Okay, we can immediately see that's a test cross. So we didn't need to know that it was homozygous recessive because that's the definition of a test cross. But they were just generous to give us that extra detail. Okay, let's say this is dad. Let's say this is mum. Okay, it doesn't really matter which way around you do it because it's autosomal anyway. But let's go ahead and do this. So when identifying recombinants, the first thing you need to do is combine the parental gametes to get a kid, okay? How that works is dad, you have to write out, if we remember from meiosis, you get 4 gametes. Okay, so dad is going to produce 4 and mum is going to produce 4 gametes, and then those combine to form a zygote, right, which is going to be these kids here. So let's go ahead and do that. So straight off the bat, this is one of the haploid gamete chromosomes the dad could pull off. If we notice, it's just off this side, the chromosomes pull apart with no crossing over, okay? That one was just this one. All I've done is separate it into 2 haploids. That's it, okay? Now we have to assume crossing over has happened for the other 2 combinations that the dad could possibly produce. So these are 2 of the combinations he could produce. The third one he could produce is that to that, okay? There's a lot of different ways you can actually draw a chromosome that's crossed over. The easiest way is just to do this vertical representation and then you draw it across, okay? You don't move anything. So this one is just going to be big B, little a, okay? I just write this one and I write this one, that's all. I don't move this here like some places show to do. I don't do anything weird like that. You just write it across diagonally. So it's big B, little a, and then the last one, again, just write it across diagonally, is going to be big A, little b, okay? Applying the same with the mum, just so we get a procedural method here, we end up with A, and you'll notice something while I do it, AB, AB. When I write across like this, it's going to be AB, and when I write across like this, it's also going to be AB, BA or whatever, AB. Okay, we notice these are all the same, so when it's homozygous recessive, you can just write 1, because that's the only combination the mum can produce. All 4 of her gametes, even if crossing over happens, are going to look like this, okay? So hopefully you're following everything so far here, and it all makes sense. Now, obviously, we need to get what the kids are going to have. So we've made our gametes here. These are the 4 gametes the dad has produced, and the mum has produced 4 of these. But just for simplicity, we're only going to write 1, because however we combine them, we're just going to be combining this all over and over again. So I don't just want to, I mean, I could write 4, but it's just using up space on my page of the same thing, right? Okay, now we combine these. So we know fertilization happens between this one and this one, between the imaginary second one and the third one and fourth one. So I'm just going to write it again. This one and this one, between this one and this one, and between this one and this one. Okay, so let's do that. So this one and this one combine. I'll draw them up here. We're going to end up with like this. Okay, this one and this one combine. Oh no way, my pen just died mid-video, bro. Oh no, it's gone. This one and this one combine, so we end up with A, B, A, B. Okay, these are the kids I'm writing now. Okay, this one again and this one combine. You see what I mean now, because I would have just written A, B again 4 times. I'd be combining that one and that one, and then that one and that one, but I'd still be combining the same thing. So I only need to write the mum's one once. Hopefully you really get what I mean there. Okay, this one and this one combine. So it's A, B, B, A. And then finally this one and this one combine. So it's A, B, A, B. Definitely haven't made any mistakes there, have I? Yeah, I think I was good. Okay, so we've got our 4 kids here, done and dusted. Kind of did it quickly, so I'm not sure if I made any mistakes, but anyway. Then we just need to look which one of these, what are the possible recombinants in this test cross, okay? So these are the possible things the kids could be, but now let's have a look at which one of these don't match to the parents, because if we recall, that's what a recombinant is, a genotype that does not match one of these parents. So these are the genotypes of the kids. Oh my gosh. These are the genotypes of the kids right here. Okay, and these, the 2 things I drew before, I'll just clean up the dad, is the genotypes of the parents. So we have the 2 parent ones here, the mum and the dad. Then we have the kids here, these 4, okay? So let's have a look. This one matches to the dad because it's big A, big B, little a, little b. So that is not a recombinant. This one, little a, little b, little a, little b, matches to the mum. So that cannot be a recombinant. This one, little a, little b, big B, little a. Just checking. Okay, that's going to be a recombinant. Does not match to either of the dad or the mum. And big A, little b, little a, little b, recombinant. Because I don't see this in either of the parents. Okay. So now let's have a look for which one is in that answer. So immediately I can see a big B, little a, little b. So this one is looking like that one. And big A, little b. Yeah, so I'm just going to confirm C, but check the other ones. So big A, B, we can see that actually just is the dad, so that's not recombinant. Big A, little b, little b, big A, little b, big A, b. Okay, those would be recombinants, but because they're not matching the parents, right? But they're not possible kids, because I don't see a kid that has big A, little b, and little b, and little a, little b. Okay, I do see that kid, so he's right here, but not 2 of them. Okay, so the thing with multiple choice is choosing the most appropriate answer, and this has 2 recombinants we've identified, this one and this one, but this one just has 2 of 1 recombinant we've identified, so it's technically correct, but not the correct answer. And this is just not a possible kid. Okay, so the answer will be C, and I haven't done this question in ages, so I wrote it down. And I think I write it down so you have to highlight it. Let's just check if I got this wrong. Oh my days. How do I Okay, C, nice. So yeah, that's the process. Let me know if you have any questions. Cheers.